Welcome to our webinar. Sharing more Allergy Public Health. I'm also involved in the Precision Medicine Resource CTSA, and that's how I'm kind of like into this. It's my pleasure to introduce Dr. Christine Garcia, who's in the Division of Pulmonary Allergy and Critical Medicine. She, as you may know, was recently named Director of the Columbia Precision Medicine Initiative, taking over from Tom Maniotis. And we've been talking to her about helping us with our precision medicine resource within CTSA as well. And so we're very glad you're going to help out with that. So Dr. Garcia received her training, her MD, PhD at UT Southwestern. She's done some really great work on pulmonary things, which she's going to tell us about today. So with that,
thank you, Gary. I appreciate it. Always fun to present to the home audience. So thank you so much for coming. And I hope this is lively and we can have a nice back and forth. Feel free to ask questions. So I'm going to tell you a little bit about the work that we've done in pulmonary fibrosis and the end kind of framework around precision medicine. If you give me that allowance. So let's see if we can start and advance. Let's there. I got it. All right. Whole genome sequencing was actually provided to Dr. Dave Goldstein through a grant through AstraZeneca, a consultant for two small companies. So I'm going to tell you about the disease itself. We're going to start with common variants, a little bit about the precursor condition, ILAs, rare variant genetics, where I'm going to spend most of my time, and then talk about telomere length as a biomarker for the disease. Talk to you about some novel discoveries and some ongoing work. So forming this is a little bit of a work in progress, and then perhaps some relevance. And this is where I'd like to have some great dialogue about precision medicine here at Columbia. So I started as a pulmonologist with a keen interest in idiopathic pulmonary fibrosis. Is a rare disease. It's more common in older people. There is no cure. Really early recognition and treatment now is the key, because the treatments can slow disease progression. Symptoms are very non-specific, with just short-windedness, especially with exertion and cough. It's more common in men and smokers and those with previous environmental exposures. And the clinical workout really seeks to rule out a lot of different etiologies until you end up with this diagnosis. And the diagnosis is made really radiographically. Well, I'm showing you a normal chest X-ray on the left-hand side that then becomes like this smattering of multiple white lines. I'm showing you a normal CT axial cut in the middle, and with pulmonary fibrosis, again, just this snowstorm of lines, honeycombing reticulations, etc. And what is happening is the fibrotic process is replacing the normal lung, so that there is a limitation in diffusion. So it's difficult to get oxygen across the pulmonary capillary vasculature, which is why short-windedness with exertion is the first symptom. So we know that from multiple different epidemiologic studies, it increases with prevalence with age. It is the prototypic fibrotic interstitial lung disease. It's also the most lethal one. It's a leading indication now for lung transplantation across the US. Medications don't cure the disease. It doesn't even stop it. It just slows progression. So we've got a lot of work to do in the pharmaceutical space. But this age associated increase in prevalence and incidence is very, very striking. And it's estimated now the prevalence is somewhere in the range of 1 to 150, 1 to 200 for those greater than 65. And its incidence is rising now. Multiple studies across multiple different countries. We don't really understand that. It could be because we're now screening more people with CT scans. So for example, in South Korea, where they screen every month, that has the highest prevalence of all different countries. So imaging is really going to be the key for making this diagnosis in the future. And I think that's part of the reason why we know that its incidence is rising. So what about the genetics, which we're going to talk about today? So it's a lovely disease to study from a genetic standpoint, because it really does have strong effects from both rare and common variants. Common variants were first discovered through GWAS studies, and really they're predictive, I think, for population-based risk for this disease. And it's very strong findings from a number of different investigators, but starting with David Schwartz's lab, found that the strongest effect, you can see this big peak on chromosome 11 is really in the promoter region of MUC5B. And then there's multiple other peaks. There's now been multiple iterations of GWAS studies. The latest one that I could find has more than 11 patients across a global biobank, more than 1.4 million controls. And you can see there's multiple different peaks now on the Manhattan plot. We're starting to see overlap between IPF risk peaks and actually COVID severity peaks, which is very interesting because, as you know, with severe COVID infection, pulmonary fibrosis is one of the most dreaded complications from that infection. And we're also seeing increased power to detect novel variants by increasing the diversity of the patients that are included in these GWAS studies. So a lot of the novel variants really driven by non-Europeans. And we know that COVID worldwide causes pulmonary fibrosis, even though the IPF studies say it's more common in those who are white or European ancestry. COVID certainly showed no mercy across people across different countries, and severe COVID infection leads to pulmonary fibrosis in people of all ancestries and across the world. But pulmonary fibrosis, or what we call IPF as a disease, is really just the tip of the iceberg, and it sits on this huge group of patients that have what we call interstitial lung abnormalities. So these are the earliest findings of fibrosis on imaging studies, and these are extremely common. So in population cohorts of people over the age of 45, it's been estimated that more than 10% of us all collectively have interstitial lung abnormalities. So since I came to Columbia, I've been really involved in the MESA cohort. And so Claire McGroder here really spearheaded a project where we looked across the different MESA exams. We excluded those with ILAs at exam 1, and then looked at the new incidence of ILAs across 12 years worth of exams. From what we could find, this is the first description of incident fibrotic ILAs. And we showed that at a rate of 3.5 per 1,000 person years, it's very similar to the incidence of COPD. So when you're thinking about fibrotic radiographic abnormalities, this is a common condition, even though those that get IPF or the full-blown disease is quite rare. We also showed in that paper there's sheer risk factors with IPF, not only male sex, older age, but also genetic factors. And that's shown in this slide. We showed that there's an association with a polygenic risk score for IPF, either with or without that dominant driver, the MUC5B promoter variant. We could show that there was an association between that and the incident fibrotic interstitial. So that's for common. What about rare? And I'm going to spend quite a bit of time on rare variant genetics. That's really what I've been working on for a bulk of my career. And it really started with families. So for those trainees in the audience, I started collecting families as a fellow. It was something that I was shocked about. I would go into transplant clinic. Pulmonologists typically don't ask thorough family histories. And so maybe I was a little bit unique because I was asking family histories. And I would find these individuals who are presenting in the transplant clinic that had just a very strong family history of pulmonary fibrosis or something that could be pulmonary fibrosis, such as end-stage tuberculosis, but every culture was negative, emphysema without any chest imaging, or pneumonia, again, without any imaging. So started collecting families. We're now, I think, past family 450 now. So these were two of the ones that we first did. And since my background was in linkage, we did a linkage scan of markers across the genome. We had a strong hit at the tip of chromosome 5p. And interestingly enough, Miri Armanios had published that pulmonary fibrosis was part of the spectrum of disc keratosis congenita, and had shown that a couple of years before. So we sequenced that as a candidate gene. We've now sequenced this gene across multiple different families and individuals. And this slide is very old. It's from 2007. However, we keep finding multiple rare variants in the telomerase, the protein component of telomerase, so the TERT gene. In those two families that I showed you, one had a frame shift, this valine at 747, frame shift right in the middle of the reverse transcriptase domain of telomerase, which is the key catalytic domain, and the other one had a mutation that was predicted to be a deleterious missense, again, right in the middle of this RT motif. But we keep finding mutations in this gene. It is now the top representation of mutations are found in TERT when you screen people with pulmonary fibrosis or familial pulmonary fibrosis. I think that this discovery also taught us a number of different lessons. Again, seen across multiple different cohorts, there are always loss of function or deleterious missense fields that we find. We find that they're extremely rare or ultra rare for these studies that we did. They were the only ones in all the databases, never found in population cohorts described at that time. And then finally, they span the entire gene. So it taught us that the gene was the unit of importance when we're looking at rare variants. And of course, the geneticists here certainly know that. And so, but that was more of a novel concept back when we first made these findings. And so I happened to be talking to Rick Lifton at a conference and telling him about this. He goes, "Oh, we just got this grant to the Yale Center for Genomics where we want to screen individuals with rare diseases, where you've ruled out other etiologies." He sequenced 100 of our well-characterized probands. We did a collapsing analysis and 2 new genes popped out using 2 different models, either the model on the left, novel damaging or deleterious missense, or the novel on the right, which is damaging loss of function. So splice site, stop codons, or frame shifts. And RTEL was a gene that had already previously been tied to the telomere pathway, but PARN at that time was completely novel, had never been linked to the telomere pathway. I'm just going to show you an example of one of the PARN families that I think is quite illustrative for this disease. We actually found the exact same rare variant. This is a splice site, the -2 position, A to G, so it's heterozygous splice site mutation in 2 different programs in the cohort that we screened. One was a patient of mine that had since died, the one on the right. One was a patient of a colleague of mine. They had the exact same variant. And at that time, the world's population databases had never reported such a variant. So when we dug deep into family history, we actually found that they had the same exact great grandmother. And even though they had lived over 200 miles apart and didn't know about the existence of the other family, they're all connected through this relative. And so we can see the co-segregation of this pathogenic mutation across multiple generations. And so what we saw is, first of all, a strong signal for this link to pulmonary fibrosis. So there was a linkage score that's not found in the general population. But interestingly enough, you can see that some of the individuals with plus signs, meaning that they had a heterozygous rare variant, did not have pulmonary fibrosis. And we had, at this point, we were just screening, do you have pulmonary fibrosis or do you not? Not any in-depth CT scan. And you can see even 60s, 70 and 80-year-old individuals had mutations. So there's incomplete penetrance. So for this disease, it's not just the genetic risk, but it's most likely other factors such as the environment or other polygenic factors that are driving the disease itself. How do we fit this into the existing knowledge of telomere-based disorders? So diphtheria keratosis congenita is an incredibly rare pediatric disorder. It's the prototype for short telomere disorders. Typically, patients have heterozygous deleterious mutations or compound heterozygous mutations. And then there's a plethora of short telomere phenotypes that they can present with. Skin manifestations are the ones that were first described. That's the name, keratosis congenita. They have a hyperpigmentation of the skin. They can have dystrophic fingernails. They can also have oral leukoplakia that has a very high chance of changing into squamous cell carcinoma. But then they also can present with extreme bone marrow dyscrasias, usually aplastic anemia requiring stem cell transplantation. And then older on, especially after transplant, they will develop other features. And pulmonary fibrosis is one of those features of the short telomere syndrome. They have telomere lengths less than the first percentile, and that's pretty much established across all the experts, and that's by the flow fish assay. So how does that contrast with the disease we study? So with familial pulmonary fibrosis, is uncommon disease clearly of older adults. It does not present in the pediatric age group. The same genes, however, are represented, even though TERT, PARN, and RTEL are the ones that are most commonly seen. Not all of the genes that are seen. DKC1, by the way, is an X-linked gene, and that causes the majority of presentations for patients with DC or dyskeratosis congenita. telomere, the short telomere syndrome features are seen, but in general, they're much more mild. Like you may just see a mild anemia, a mild thrombocytopenia. You might have liver dysfunction, but it might be not like liver cirrhosis that you might see with DC. And the telomere lengths that we are seeing, a cutoff less than 10th percentile for age-associated lengths, and over 85% of those patients with mutations. So there's a lot of features shared in common across these diseases. Interestingly enough, we see both rare variants, but also these common variants were found in the GWAS hits. So a great example here of telomere pathway variants and genes seen across, really speaking towards the critical importance of the telomere pathway as being part of the underlying etiology for pulmonary fibrosis. So telomere attrition, in my mind, is one of the key features for large subsets of patients with pulmonary fibrosis. And just to remind you, the telomeres are the ends of the chromosomes. Of course, we have multiple different cells represented in. So when we're making a measurement of telomere length, we're really just measuring an average telomere length across multiple different chromosomes, across multiple different cell types. And so for all of these assays, since telomerase is limiting for us as humans, and we can argue about why we think so, but you know, many people, including Jerry Shea, thinks it's to limit our likelihood of having cancer at older ages. So TERT is limiting, the expression of TERT is limiting in replicative cells. So we see a decline in telomere length over age. So in the middle panel, I'm just showing you telomere length data for individuals with a wide range of age. So those were our normal set that we've used across multiple different assays. Here, the Southern blot assay, but we've also used it for a qPCR assay, and we see the same thing. Flowfish, same thing. So if you see on the right-hand panel, this is where I'm showing you telomere length. Each individual dot is a proband. So you can see for all probands of those with familial pulmonary fibrosis, some of them have quite a bit of extreme telomere shortening, less than that 10th or 1st percentile bar. But then some of them don't. So you can see there's some that are in that normal range, that aqua colored normal range, but then there's some that are in that shorter range. And then when you really map on top of that, those with these rare mutations, like I'm doing here on the right-hand side, each dot is an individual now across multiple different families where each individual person has a mutation, one of these loss of function or deleterious missense mutations in TERT or PERT. And you see all of them across the age spectrum tend to be less than the 10th percentile, but pulmonary fibrosis is really just seen in those that are older. So again, there's something besides the genetic risk that really gives rise to this disease, even in those that have these, what I would say, high effect size rare variants. So age plus the genetics gives rise to the disease. So we started first studying those with the rare manifestations of telomere mutations. And one of the striking things that we found is that the clinical diagnosis of these individuals was not restricted to IPF. So on the left-hand side, I'm just showing you a schematic that's kind of just visually telling you that of all the people that had these rare mutations, half of them had a clinical diagnosis of IPF, but then the other half had a variety of different diagnoses. They, you know, pulmonologists like to be splitters, I would say. And so they had a bunch of different diagnoses like chronic hypersensitivity pneumonitis or idiopathic interstitial pneumonitis, all these other type of abbreviations. But if we looked at those individuals and we looked at the survival curves of them, we said regardless of which gene was involved or regardless of if the diagnosis was IPF or non-IPF, which in most patients portends a much better survival than IPF, we saw that those curves were pretty much superimposable. And so the genetic diagnosis, I think, is correlated to a wide heterogeneous number of clinical diagnoses, but it portends a uniformly progressive type of disease that's quite lethal. When we also looked at these patients, you know, you guys remember that there are promoter variants in the promoter of TERT which are the most common alteration in cancer, seen in over 90% of bladder cancer, different types of brain cancers, et cetera. So we looked specifically and sequenced the promoter region of this niche TERT in people that had these mutations. And what was striking is that even though we didn't see them in skin or in buccal cells, we would see that about 10% of them would pick up the acquisition of a somatic mutation in these TERT variants in the blood. And there were a couple that came up over and over again at the -124 position and the -146 position. And then we looked to see in phased chromosomes, if they were in phase with the wild type or if they were in phase with the mutation, because remember, these people are heterozygous. You see on the right-hand side, it's more typical that these promoter somatic mutations were in phase with the wild type, shown as the open boxes, not the red boxes. Occasionally, you would see one of those promoter mutations in phase with the mutation, but in general, we saw it phased in the same phase as wild type. We did additional studies here to show that the acquisition of these promoter mutations actually caused an increase in telomerase activity cells, which is consistent with the literature, consistent with the fact that they are found in many different cancers. In fact, these might be the most highly represented mutations found in cancer. So putting all of that complicated work in kind of in a schematic diagram, what we see is that in mutation carriers, telomerase is upregulated. So there is this balance. So since the patients themselves are heterozygous for loss of function mutations, so they have less telomerase, there's a predilection for senescence to overcome. But over time in these patients, there's the acquisition of promoter mutations, and at least 10% of it insists with wild type. So again, that pathway is associated with cancer. So there's a yin yang here that we're seeing in patients, especially those of older age. In studies in dyskeratosis patients, we don't see this in the pediatric group. We really see this in the older patients, especially over age 50, this acquisition of somatic variants. So what about telomere length itself? So here we studied, and we first showed in 2014 that telomere length itself was a biomarker that was predicted of a worse survival across all comers, not just those with mutation carriers, but all of those with IPF. Multiple different authors, I've listed the references here, have shown that across multiple different cohorts. And this was just one of the last ones that was published. We partnered with Deji Adesoya, and he showed beautifully the increase in hazard ratio of worse survival the shorter your telomere lengths are, and plotted those survivals based on whether they were shorter or greater than the mean for age-adjusted telomere lengths. He also showed basically, regardless of that clinical diagnosis, IPF or connective tissue disease associated pulmonary fibrosis or hypersensitivity pneumonitis, all of those, there was worse survival with telomere shortening, and across all ethnicities as well. So this is not something that's just particular to one thing. We are seeing this across multiple different cohorts. And another interesting finding that we thought about, since it's predictive of survival and clinically, we had known, or I had personally seen patients with these mutations that after they had been exposed to immunosuppressive medications, they had a high likelihood of having sepsis and dying. So we surmised that perhaps this biomarker was interacting with medications that patients were given by their physicians. And really the best resource for testing that hypothesis was a clinical trial called the Panther trial. For the non-pulmonologists in this audience, this was really one of the first well-done randomized clinical trials in which patients were either given a combination of prednisone, azathioprine, which are immunosuppressive medications, and NAP, or randomized to placebo. This was the first trial that showed definitively that this therapy, which according to experts at that time was considered triple therapy for the disease, caused more harm. So the y-axis shows a probability of death or hospitalization. So this definitively showed we don't use those medications now for people with IPF. But it took us some time to get our hands on the samples because what we really wanted to test was really here. So of those that were enrolled in the Panther trial, and for those that consented for genetic studies and were given a sample and DNA was available, could we measure telomere lengths and then see of those individuals who were randomized to treatment either to triple therapy or placebo, could we see a difference that would basically explain the outcome that I just showed you of the New England patient? So it took us, one of the people to ask me how long it took me to get collaborative. So it took me 8 years to get my hands on these samples, but they are available through BioLink and through collaboration that we developed. We finally got our hands on them. And here we measured the telomere lengths, and I'm just showing you the data split too. On the left, all the patients with telomere lengths less than the 10th percentile. On the right, all the patients with telomere lengths greater than the 10th percentile. And then we're showing you the survival curves stratified by whether or not they were receiving triple therapy or placebo. And you can really see that the adverse effects are really just seen in those with the short telomere lengths and those that are exposed to medication. So it's this interaction of a telomere length with the immunosuppressant, which is driving the signal, I think, in the Panther study. There was a study that was done simultaneously that we could get our hands on BioLink, the NIH BioLink repository. That was called the ACE study. So even though it was studying warfarin's effect, patients were similarly either on prednisone and azathioprine or a placebo. And we looked in that study and we saw the exact same things. So here, I think what we're seeing is really telomere shortening. It's predisposing patients to this immunodeficiency phenotype, which is part of the short telomere spectrum. And then that's being unmasked when they're given these doses of immunosuppression medications for almost up to a year. So pretty long exposures to suppression.
But even in the one that they have over 10% is effective.
I'm sorry?
Even in the one that they have over 10 percentile, still ineffective. So you should.
Right. It's not effective therapy, but they're not having the same harm, right? Death and hospitalization. Yes, exactly. And so recently, we collaborated with a group, and this study was really spearheaded by Chad Newton, where we looked at the same thing in patients not with IPF, but basically with non-IPF pulmonary fibrosis, all those other types of diagnosis. And in the discovery and replication meta-analysis, we're seeing that same trend. Those less than the 10th percentile, we're seeing that there's an increased hazard ratio now for harm in those treated in this way. And we do think that this would be an important biomarker in the future, because many times as clinicians, we don't know what's the appropriate first line therapy for those with these unusual diagnoses like hypersensitive pneumonitis. And if we had easy access to this measurement, it could help stratify clinical management of these patients. And David Zhang was involved in this as well. And really, we just see it skyrocketing up with increased hazard ratios, the shorter the telomere length is. Yes.
Do you see any bone marrow suppression?
So a little bit of anemia, sometimes thrombocytopenia. Yes, we do see that full spectrum of short telomere phenotypes. They just don't tend to be as dramatic as what you'll see with DC patients that'll present with aplastic anemia. So this is something that's ongoing, and I'm not going to really tell you much about this, but we're trying to leverage these genetics discoveries in the clinic here at Columbia. And there's ongoing studies now that we're trying to see how it might impact our clinical management, especially telomere lengths for these patients with these rare diagnoses. And for the rheumatologists in the audience, rheumatoid arthritis, scleroderma, et cetera, there is a subset of those patients that are older that present with pulmonary fibrosis. And so we're investigating certified telomere lengths, also genetic sequencing for all of those family history and short telomere lengths. And we're doing cascade testing at the end. So more to come really on how we're leveraging these genetic discoveries. But of course, the key is going to be, how reproducible this is across multiple clinics. Okay, so with that, so those are the telomere related stories. But as I showed you in that plot of patients with familial pulmonary fibrosis, not everyone had telomere shortening. So we know that there must be some non-telomere genes related. So one of the first things that we did, David Zhang and I came from UT Southwestern, we brought our cohort, we got sequenced here at Columbia, It went through IgM, it got merged with all the exome data that was here in the IgM, and we did a collapsing analysis now with large numbers of controls, large number of cases, and we found a new gene called KIF15. And we could see that signal both in the deleterious rare variant model as well as protein truncating model, and without a signal seen in the synonymous variants. And we used a replication cohort from dbGaP that had been released in the meta-analysis. Now we're seeing very, very strong odds ratios for deleterious or protein truncating variants in some of these genes, but the KIF15 ones were also above genome-wide significance. Similar to the other stories, most of the signals being driven by those with strong family histories of pulmonary fibrosis, as you would expect, or rare variant genetics. So in our cohort, which is enriched for those with families, we see a stronger effect size. In cohorts where it's more sporadic cases, we see fewer number of rare variants. And this shows that in telomere length here, I'm just plotting it in terms of the observed minus the expected. Those patients with KIF15 rare variants don't have any telomere shortening, like in contrast to those with telomere-related mutations. KIF15 common variant had been linked in a previous GWAS, and the variant was in between KIF15 and the next gene. And David did fine mapping of this locus and actually found a variant that was in very strong association with pulmonary fibrosis that sits right in the middle of KIF15. And he showed replication, he showed it both in the discovery cohort and in the replication cohort. And strikingly, KIF15, the signal is both now in rare variants and in common variant. So this is a mitotic cell division pathway, I'm calling it, because rare variants in another gene, spindly1, which is important for spindle and cell division, are also found. So it's a non-telomere mitotic cell division pathway that's important for pulmonary fibrosis. So KIF15 is basically a protein that interacts with kinesin. It's very important for sliding the microtubules apart to promote spindle elongation and allow the cells to divide. Interacts with Ki67 and yeast 2 hybrid cyst screens. In our functional studies, we showed that we have to inhibit its partner, EG5 or KIF11, before we can elicit the phenotype of cell death. And here in HAP1 wild type cells, when you give the EG5 inhibitor, there's no difference. In cells that are deficient in this gene, so HAP1 knockout cells for KIF15, when we give it the inhibitor of its partner, EG5, only in that case do we see a reduction in cell growth that can be partially reversed by expressing the full-length wild-type KIF15 gene, but not the truncated mutations or the mutations that we found in patients. Different concentration curves are shown below. We also similarly showed a cell growth phenotype in cells that were heterozygous or homozygous for that common SNP that was linked to the disease itself. So there's some striking similarities, I think, between this pathway and the telomere-based pathway, because it's both rare and common variants linked to disease, loss of function variants enriched really in our familial kindreds, common variants seen in the sporadic cohorts. Patients that have homozygous pathogenic variants really are presenting with pediatric, extremely rare syndromes. So dyskeratosis congenita being the prototype for the short telomere syndromes. There's a Bradley-Carey syndrome for those that are homozygous, loss of function mutations of KIF15. It's mostly a neurologic syndrome found in very young infants with microcephaly, but there's also, interestingly, a bone marrow phenotype of thrombocytopenia. Both are necessary for cell division. They're both expressed in dividing or replicative cells. Telomere dysfunction has been shown in mouse models to be important, especially in the epithelial cells in the lung. David showed that KIF15 is expressed in the epithelial cells and may be different to different levels. And those with IPF had reduced expression in epithelial cells. So again, pointing towards an epithelial problem. So this summarizes what we know about genetics of pulmonary fibrosis. Those with familial disease, I would say that our hit rate when we send for genetic sequencing, or if we do this in the research lab, is about 25% with familial pulmonary fibrosis. So we have more work to be done. Those with sporadic disease, really, we're only seeing 5%, but we're seeing the same exact players, the same exact genes. And I would say that, you know, of all the families that I've collected, the one that has the largest number of affected individuals, it has 19 affected individuals and it spans more than 6 generations. I still have not figured that one out. And that's a short telomere, autosomal dominant, incomplete penetrance, pulmonary fibrosis with thrombocytopenia phenotype. So there are certainly more genes or structural variants or non-coding variants that are perhaps linked to these that are yet to be discovered. So kind of just, you know, the starting point for etiology of pulmonary fibrosis in the sporadic forms. So hit rate of 5%. Okay, so now I was just gonna, I went maybe too fast, but I was gonna just tell you about some of the ongoing work. We've partnered with people here in the room that were looking at models to try to better understand the mechanism. And again, this is completely work in progress and a little bit out of my wheelhouse. So it's things that are taking us maybe a little longer to do, but certainly something that I feel strongly that we want to continue to push forward. So we've had this mouse model now for a while. We used CRISPR to basically put in a point mutation. So, you know, besides the telomere related genes, the KIF genes, surfactant A was one of the first genes that we discovered in the lab, and really just in 2 families. So, and with all the sequencing, we know it's one of the rarer forms of pulmonary fibrosis, but it's very penetrant and it's a dominant form of familial pulmonary fibrosis. And interestingly enough, the patients have this very high risk of lung adenocarcinoma, which has been described in the disease, but especially for those with surfactant mutation. So we made this CRISPR model where we just introduced the point mutation. And as predicted, that mutation in the carbohydrate recognition domain basically causes protein folding. The protein gets stuck in the endoplasmic reticulum like we had shown in cells and has difficulty transversing through the cell and being secreted out into the alveolar space. And so the homozygous mice here inside the lung, you can see less mature SPA in the homozygous mice lung, but out in the bronchoalveolar lavage, there's no SPA, but you can see amounts of surfactant D and other proteins appropriately secreted. The mice are viable in the homozygous state, and at baseline, without any injury, there's a little bit more cellularity. There's a little bit more type 2 cells that we can see by counting visually or by flow cytometry, but there's no overt problem. They live past 1.5 years with no spontaneous pulmonary fibrosis. However, with injury, and the injury that we've really used is this week-long infusion of bleomycin through a pump, a mini pump that's implanted under the skin, we can see that there's an increased risk of death over time, and that's really driven, especially by the male mice, both heterozygous and homozygous, in contrast to the females. We also see that there's the appropriate pulmonary physiologic impairments that we would expect to see after this type of injury, where there's a compression of the PV curve, so the lungs are getting much stiffer. And again, this is much more dramatic in the males than the females, and significant deformation of the PV curves in both the heterozygous and the homozygous mice. And remember, the humans are heterozygous, but the mice are viable in the homozygous. And then when we look histologically, we can see lots of fibrosis, but again, especially in the heterozygous and the homozygous mice. Jahar Bhattacharya's lab and Naheem specifically, who's here, has shown us that there's increased susceptibility to infection, as would be predicted if the mice cannot produce SPA in the bronchoalveolar lavage. And we think it's a great model, especially because we have this male dominant phenotype, which, as I told you, is something that is seen in humans as well. So we've looked to try to figure out, well, why might this be? And really, we struggled for a while, and then I'm just presenting some unpublished data that David recently has done. David Zhang, who's a whiz with bioinformatics analyses, and specifically single cell analyses. So what we did in the lab is we used the hashtag technology to prepare epithelial subsets of the lung, and then individually label them with these tags, so that we could label multiple different males and females, and then submit that for single cell sequencing. And then on the back end, on the bioinformatic end, use the tags or the hashtags, then deconvolute the data to those that had the mutation and the gender, et cetera. And I'm just going to show you the aggregate data because we're just still diving down. And what we're seeing strikingly at day 35, so at a point in the injury where there is a lot of fibrosis, we're seeing dramatic differences in the numbers of these subtypes of type 2 cells. So we see greater numbers of basal cells in the mutant mice than wild type. We see a tremendous decrease in the number of activated type 2 cells. And then there's trends of fewer PAT cells, which are transitional cells after injury, which are similar to senescent cells that you see in the human disease process. And we're seeing fewer type 1 cells after injury. And so what we're doing right now is looking at the temporal and spatial changes of these cells after injury and determining which gene expression patterns are associated with these changes in cells. And I think that may be the picture here that we're seeing with this particular mutation, is that it's causing problems after injury in the dynamics of how the cell state exists. And again, ongoing work, and I'm kind of glossing over it because it's still work that's in progress, led by Tana Grong in the lab, as well as many others. We just started with our KIP15 mouse model. Turns out KIP15 knockout mice are available. We got them ripened, they're viable, fertile. And we are just as a first pass, we just looked to see, again, using this hashing technique, if there are differences in numbers of cells. And strikingly, what we see in the lung is that there are more monocytes, classical monocytes, more epithelial cells in the KIP15 knockout. And now we are doing the similar type hashtag analysis after bleomycin. And we see the same trends, greater number of basal cells, fewer number of activated type 2 cells in the KIP15 knockout mouse after bleomycin 35 days later, fewer type 1 cells. So again, replicating, trying to follow in the same vein as what we did with the surfactant mouse, trying to see what that means and whether or not it's it is somehow telling us more about the biology. And I have to say that, a lot of work in pulmonary fibrosis, at least the mouse models, has shown that the 3 key cell types are the epithelial cells, which we know from the genetics and the surfactant mutations of telomerase and KIP15 are key for driving the disease, but also monocytes. And then finally, the effectors of the disease are the fibroblasts. And so I think the intersection of those 3 cell types are going to tell us something here. Now we're working with Donna Farber's lab and specifically Alex, the graduate student, to really look at flow cytometry of the monocytes to see which type are different in the different states at baseline. So with that, I think pulmonary fibrosis, at least for me, the way I see it, is a prototype for precision medicine, but you can insert many different diseases in that. And I think that, what's great about being at Columbia is so many examples here exist for various diseases, and certainly in this audience, many are represented. And so, it allows us to use genetic discoveries to improve our understanding of disease. And I think more particularly, it allows us to really hone in on well-phenotyped patient cohorts and understand more about the biology, not just those with familial disease or strong family history of disease, but how does that tell us about the biology of sporadic disease? And then more broadly, population-based studies, risk of disease on a population level. And I've just told you one story, but I know multiple stories exist here in this audience. And we want to make sure that we can promote those types of research here at Columbia through the Columbia Precision Medicine Initiative. And how can we do that? Well, we certainly have to leverage cutting-edge technology. We have to be using state-of-the-art sequencing and bioinformatic analysis. I think, I just told you the KIP15 story, and here, really that's a story of leveraging the Columbia-specific repository of control samples, over 26,000 samples that gave us that signal to pull KIP15 out of the noise, because genetic discovery work is very messy. It's very noisy. And so we always need better methods and tools of pulling signals that are real out of the noise. And so I certainly think that we need to make sure that we can provide these resources across the entire Columbia community, both from the sequencing point, but also the bioinformatic analysis, and specifically large data sets of Columbia patients that are well phenotyped, for us all to do this, and then explore how genetic biomarkers may improve or impact clinical care, right? Because that's all what we're trying to do. We're trying to improve clinical care. And how can the knowledge that we gain improve that in a real way? And then again, as a side note, I think, not all of these things are available to be studied in different models. As we all know, some models don't completely replicate the human disease, and certainly for age-associated diseases of adult individuals, it's very unlikely that we're going to find a nice, clean model that exists. However, we have to leverage the models that we can make to try to glean some understanding. Certainly, fish don't have lungs, so they have gills, they don't have lungs, so I can't make a zebrafish model, right? And so I'm limited in the models that I need. Certainly the larger animal models like pigs could really replicate more pulmonary fibrosis, but those are things that are beyond the range of what, we could do on a single, from my standpoints. But we need to develop the cellular and animal models. And here, Hans has a beautiful setup, and he's actually collaborating with us with the KIP50 knockout to look to see how iPS cells may also tell us more about the disease. So I think we need to collectively use our tools across multiple different ways to see how we can use those in a bigger way to understand disease. So this is just a summary. I think I've kind of gone over everything, and a huge thank you, especially first to the patients and families that I've partnered with over the last almost 20 years now. The ILD clinic here that is just an amazing powerhouse of physicians seeing patients. The team in the lab, really thank you. Huge thank you for the ongoing work. Collaborators, ongoing projects, collaborators in past projects, really could not have done it without our collaborative effort and the funding sources. So with all of that, I thank you very much for your attention, and certainly turn it over late in the day for questions and comments. Hey.
Can you elaborate between the telomere length as a biomarker or as a sort of consequence of the disease?
Certainly. So I think it's with the rare variant telomere mutations. I think it's the driver that's for showing us the short telomere lengths in those patients. When we look at families that have the mutation and other in related individuals that don't have the mutation, there's a clear separation in telomere length. However, in the entire disease population itself, we see telomere shortening over and over. So telomere shortening has been associated with smoking, been associated with a number of the risks that are shared in common with pulmonary fibrosis. As you know, there's an age-associated drop in telomere length as well. So I think it is a manifestation of the underlying genetics of those individuals, but I think that it also, telomere shortening may be a marker of individuals based on their environmental exposures, or, the combination of common variants that they've inherited, as well as the set point of the telomere length that they're born with. So there's a lot of great biology of telomere length. And if your parents just happen to have a shorter length, that short length is inherited by the progeny. So with a set point, plus also how much telomerase activity. So to me, it's it's fascinating topic.
So this was really great, but actually the question I have is relates exactly to that. Telomere length as a quantitative trait, has it been studied in terms of like formal heritability estimates? To what degree has it been used in discovery studies as a quantitative trait, like in GWAS or even pathway-based studies?
Yeah, so lots and lots of literature, a lot of GWAS studies on it, a lot of UK biobank studies looking at the phenotype. In fact, pulmonary fibrosis is one of the dominant signals now. They look at the data across the UK biobank. In fact, that data just dropped. They just did telomere length using qPCR assay across 500,000 people, and then looked at phenotype. So pulmonary fibrosis is one of the very top signals, but they're also seeing blood signals as well in the UK biobank. But just a fascinating biomarker. And they've looked at, the genes that are associated and same type of overlap of genes that we see for dyskeratosis genetic. And the common variants, some genes that are not really well understood also being part of the of the genetics of tumor length.
So I assume the third promoter mutations that you talked about are seen in the bone marrow, but not in the lung.
Right.
What-
So we don't know yet about the lung.
Would that maybe be a therapeutic target, or would it come too late?
Yes.
Be too dangerous?
Right, I know. So lots of great questions. Some companies are working on that right now about whether or not you can actually express it in the lung safely. The biggest concern is lung cancer and lung adenocarcinoma because of the link with IPF and lung adenocarcinoma. These patients are dying if they have a very aggressive, progressive pulmonary fibrosis. So, would that somehow be a therapeutic option, I think is unanswered at this point, but certainly interesting. The somatic variants, we don't yet know exactly in the lung how many we see. We also don't know yet of those because these are pretty rare. We're only seeing them in the rare carriers and only 10% of those. Whether or not that increase in telomerase activity somehow protects them from the bone marrow failure part of the short telomere syndrome, which is what I've really tried to look at with a lot of depth and flow cytometry, certain cell type, et cetera. But we haven't yet been able to tease that apart, but certainly within the realm of possibility, I think.
I was very curious about talking about the biology of the telomere length, the telomerase or so. I was intrigued by the data about the dexamethasone and what is the effect of the immunosuppression in the telomere length and the effect what is being seen. I just cannot put 2 + 2 in here.
Right. So we've looked, we don't have thousands of samples, but we've definitely looked at our data using the qPCR assay on patients, say before transplant and then after transplant, because all transplant patients undergo tremendous amount of immunosuppression with 3 arms of immunosuppression medication. We don't see, robust signals of telomere shortening within the time frame that we can see, but certainly medications and drugs could be affecting telomere length. It could be. But remember, those DNA samples from the studies that I showed you from Panther and ACE were obtained before they were basically randomized into getting those treatments, which is why that data is so clean and so valuable, because those samples were obtained first. They were then randomized and then the outcomes were collected. So I don't think it perhaps is causing what we're seeing in those clinical trial data, but I do think that perhaps it could be something that's yet to be explored, and something that I've encouraged my lung transplant colleagues, of which one of whom is in the audience, we need to explore that too, because if we know that those with the short telomere lengths are progressing faster, they tend to be the younger patients, at least the mutation carriers, they're progressing faster. So they're enriched in those patients that we are referring for lung transplantation. And so since immunosuppression is key for lung transplant, I think centers like our center, which does so well, because the clinicians have such a high, threshold for treating infections, getting them admitted, doing the appropriate workup. These clinicians are really amazing at how responsive they are to the needs of the post-transplant patients. But maybe there needs to be different thresholds for immunosuppression targets. Maybe there can be precision medicine approaches in the post-transplant group. So that's something that I think a number of people are interested in, including Salim Arjusway, who runs our transplant.
All right, I have one more question. I understand the bone marrow phenotype. I don't quite understand why short telomeres affect the lung, especially, working on liver. I mean, regenerates, and there's actually, I think, a protective mutation in the telomerase gene for alcoholic liver disease, but there's nothing else. Obviously, the third promoter mutation is very common then in cancer. But why the lung? Is it a functional reserve that's so low, or do you have more cell turnover than
Yeah.
I think there is?
Huge question, $1 million question, I think, in the field for the last 20 years. Why the lung? Why is the lung so susceptible? I don't know that I have any great insight. I mean, I think Paul Walter's mouse studies where he knocked out TRF2, one of the components in the different cell types, showed that it was really only the epithelial specific CRE that gave rise to the increased fibrosis in mice. He knocked down fibroblasts, they didn't see that. So it seems to be an epithelial. Then in the, you have Wellington right here. There's many different subtypes of type 2 cells with regenerative capacity and Hans, right? So John Wen. So which of those subtypes are dependent on telomerase in the human? The mouse is totally different because the mouse expresses TERT all over the place. Whereas if you look at single cell data of human lung, you can't ever find this TERT transcript. I mean, we've looked a lot and we can't find it. But in the mouse, it's easy. All those cells express TERT to a high degree. They all have very high telomerase activity when you look on trap assays. The human is very different. It's very restricted. No one really yet knows exactly which subtype of regenerative epithelial cell is dependent upon it. I think that's the limitation in the field, and maybe my colleagues will help figure that out.
Just, again, what about mesothelium? Is there any association between mesotheliomas, any of the things in mesothelium? Because of one of the possibilities is that the lung is probably one of the organs that have the greatest mechanical strain than any of the other organs. And given that it's subjected to such a thing, I was wondering if the only thing that I can think of in the lung would be the mesothelium, that it is as.
We don't see any signal with
Yeah.
mesothelium.
Okay.
Not that we've seen. That's pretty bad.
I have a
Yes.
Question from online, from Stavros. Says this was fantastic. Patients that the mutation on telomerase is inherited of many generations. You find it earlier onset. I imagine that these starting shorter telomeres.
Yes, and I glossed over all of that. Thank you, Stavros, for reminding me. There is genetic anticipation. So Mary Armanos has really shown that beautifully in her disc keratosis congenital kindreds, for the older generations with pulmonary fibrosis, the next generation, you have bone marrow failure, next generation is full of bone disc keratosis congenital. There's clear evidence of genetic anticipation. We do see that in some of our kindreds. It's just not as striking as the DC kindreds that she studies across the entire age spectrum. But yes, there is, it goes to that, what we're talking about, born with a shorter set. Thank you, Stavros, for reminding me. Yes.
Can I just ask a question about the mouse model? So when you look at the epithelial cells, you're doing flow to determine the number of epithelial cells. That I do.
So there's a number of ways that we're doing it. The data that I just showed in terms of the single cell, we're doing an epicam positive cell population from the lung and then collecting that, tagging that and sending that.
So with bleomycin injury, there's a real clear pattern of, you know, gene function. They're injured and when they start to go through the transition. So I'm just wondering if you're able to genes during this process to see if you disrupted some pathway of repair.
That's what we're looking at right now. That's what's ongoing.
Okay, and do you have any chance to do lineage tracing? Maybe cross them with.
So we have the ROSA SBC CRE. And so what we're looking at now is those ROSA SBC CRE mice that have the homozygous mutation of the CRISPR surfactant and then we hit them with bleomycin. And interestingly enough, that expression of ROSA must give it a double whammy for those type 2 cells because we started with our 100 units of bleomycin, all the males died. We dropped it to 80, all the males died. So now we're down to 45 units of bleomycin and just a couple of months. You know, I think only one has died yet in this. So we had to really decrease our dose of bleomycin in the ones with the SBC expressing TD tomato in the type 2 cells. So we're going to do lineage trace with that at different time points. We're harvesting mice at 5 different time points now. So all that's ongoing really to look at the temporal changes. Great questions.
Any intersection with the clonal? Yes.
The chip genes. So we do see those in the rare variant analysis when we do classing. We'll see some of the common chip genes. I don't know if that's because our cohort's older or because there's enrichment for some of those chip gene mutations, but they're commonly at the top of our class.
I think if there are no more questions, I'll go ahead and if we have more, maybe follow up.